I will send you some textbook pages, 4 sets of pages in the textbook that are related to classification. And these include all life forms, microbes, plants, and animals. There are a lot of pages, but those pages have a lot of pictures. So don't be too worried about the numbers of pages that you will see. Anyway, that will be in the announcement that I will send you later today when I send you the recording of this session.
Professor?
Yes.
Quick question, Alex Allos.
Alex, okay.
When is quiz 2?
Wednesday.
Wednesday?
And again, it will be a 15-minute time limit. It's worth 10 points, and it'll be available to you all day on Wednesday.
And what's it based on? Like all of last week's slides?
Yeah, based on everything since quiz 1. So it includes cell structure and function, life in the universe, and classification of organisms, which is what we'll talk about today, and what I'm giving you page numbers to read in the textbook.
Professor, I have a small question.
Your name?
Oh, my bad. Yusuf Faraj.
Yeah, what's your question?
So the worksheet, cell structure and function, that won't be graded?
That's correct.
Only the chemistry paper will?
That's right.
Okay. Because I'm just checking Canvas. Thank you.
Okay. Anything else?
Yes, Professor. You said the sheet that won't be graded is just for us to work. It's this worksheet for us.
Yeah.
Will any of the information on that worksheet still be on a test or quiz?
Not directly. There might be something related. So the purpose of the worksheet is just to help you think about the material, be comfortable with it, but there won't be a direct question from the worksheet.
Okay. You know who I am, Yolanda.
Yolanda, yeah, I got you.
Okay.
Professor, this is Alicia Carroll.
Uh-huh.
For the worksheet that's due about chemistry, do you want us to answer in each of the grids, or do you want us to list it out underneath for all the 16 questions?
Say that again.
On the chemistry worksheet, there's 16 questions. Do you want us to put our answers in each of those squares, or would you rather us list it on the bottom?
Now put them in the squares. There's enough room in each of the little boxes to write your short answer in each box.
Okay, thank you.
Professor?
Yeah.
That is Rasha. For the quiz, every week we do on Wednesdays.
Every week we do what on Wednesday?
Last week we do the quiz on Monday.
Oh, yeah.
This week on Wednesday?
Yeah, that's correct. And so there's no regular occurrence of quizzes. Quizzes will show up whenever it seems appropriate to me based on the subject matter. So the quiz 2 coming up on Wednesday covers those 3 topics because they're sort of grouped together.
Okay, thank you.
Professor, I have another question.
Yeah.
Last time I took the quiz, I felt like 15 minutes was really short. So is that going to be the same time limit again?
Yes.
Okay.
I sent you another reminder of my policy on time limits and quizzes and tests. I hope you all saw that. I sent it early in the semester, and then I sent it a couple of days ago, once again, just to remind you so you know what it is.
I have a question. Where is the new worksheet at? Because I don't see it on Canvas.
Your name?
Kiana Burns.
Deanna Burns, okay. You don't see it?
No, I
Some depends on where you're looking. It should be on the home page. It should be under assignments. If you click on that. If you
yeah,
don't
it's
see it
not
there,
under.
you might find it in files. Sometimes if you don't see it, there's a little arrowhead that you can click on and that will show it.
What is the worksheet called?
No, there are two. One is called Chemistry for Biology, and the other is called Cell Structure and Function. And the first word in each case is worksheet.
Yeah, I don't see it in my files or my assignments.
Kenya, just to chime in, I've seen it under modules.
Okay, I'll look for it and then just before class is over, I'll let you know.
Yeah, yeah. In this case, in cases like this, it's probably better to ask a classmate than to ask me. Right, I'm going to share my screen with you. Classification of organisms. Anybody not see that? Okay.
Oh, I can see it.
Good. Okay, how many different types of organisms are on this planet? Life is diverse. Over 1.8 million species have been identified to date. There's a comprehensive list called the Catalog of Life, and here's the website for it. Exact number of species on the planet is not known. In fact, we're way off on the estimate. About 92% of known species are in this catalog. So new species continue to be discovered all the time. If you look at this pie chart, you can see that in the blue sector is species that have been described and cataloged. The red part is species that have not been described, they're not cataloged, and that's about 86% of the predicted number of species. Only 14% have actually been described and cataloged.
Real quick, and is that, and this is Anthony Imolo, but is that for the entirety of the earth, like oceans, mountains, lava, stuff like that included?
Yeah, that's all habitats on Earth.
Another question, how do you know how they predict that? Like how do they make that number up? You know, like is there a process to that or?
Yes, and it's called, well, there are a couple of ways of doing it. One is a complicated algorithm that predicts based on history, predicts future. The other way to do it is what's called metagenomics. And that means that you sample the environment. So you can take a sample of air, you can take a sample of soil, take a sample of water, whatever is in the environment that you're interested in. You take a sample and then you extract all the nucleic acids. So you don't care about organisms, you extract all the nucleic acids there and sequence them. And then by matching up sequences, you can make predictions about organisms that must be there, but you can't grow them, you can't see them, you can't find them. You just know they're there.
Awesome, thank you. That's really interesting that people even figured out how to do that.
Yeah, and we'll talk later today about the sequences that are used to do this.
Professor?
Yeah.
This might be a
Ms.
dumb
Martin?
question, but what you just said made me wonder. I thought there was still like a big part of the ocean, like not explored at all. That's correct, right?
Yeah, there are parts of the ocean that are not explored very well, but there are a lot of people that go around various places in the ocean and go very deep in the ocean and take samples and then sequence the DNA in those samples.
Oh, okay. That makes more sense.
Are certain parts of the ocean just not accessible? Could that also be another factor?
Well, your name again?
Nicholas Gonzalez.
Gonzalez. In principle, there is no part of the ocean that is not accessible, but some parts are much easier to reach than others. So very deep, 3 or 4 miles down, the pressures are so enormous that it's hard even for machines to operate in those depths. But there are machines that will do it. And I think I have some pictures of organisms from way down there. And here, just out of interest, I've got some pictures of large species. These were not detected by metagenomics, but simply by careful observation. Here's a hummingbird that was discovered in 2009. Down here, lower left, is a type of sea slug, newly discovered in the Philippines. This weird looking thing here is actually a fish, described in 2009. And it seems to make a living by mimicking a coral. And this giant salamander is not a new discovery, but this behavior is new. It's the first image of one of these giant salamanders trying to eat a snake. The disturbing factor. Yeah.
I have a question about the animals. No? Do we have any evidence then we have animals from the, like birds for the prehistoria, or do we have any evidence of the birds for 100 years before now? Do we have
the fossil record
they're
is
still alive?
Right. The fossil record, paleontology, is a good source of information about species and species that are now extinct. And again, either by looking at the sizes and shapes of bones or taking DNA samples from bones, either one gives us insight into species that once existed but no longer do. And it turns out that species are born and become extinct all the time. Since the beginning of life on this planet, species have come into existence and species have become extinct as the environment has changed. It turns out that the rate of extinction is much faster now than the rate of new species coming into existence. So if you look at this graph here, starting in the 15 and 1600s, and you have mammals, birds, fish, reptiles, and the dotted line is background. So what is background? That's what I've just been talking about, the natural rate of extinction. So the natural rate indicated here is about 1 species per every million species every year goes extinct. So for every million species, 1 of them goes extinct per year, naturally, just because of changes in environment. But the other lines, the colored lines, show actual rates of extinction. You can see that they're fairly low, close to the baseline for hundreds of years. And then starting with the Industrial Revolution in the 1800s, the curves have shot up because as humans have taken more and more advantage of the planet, they're putting pressure on various different kinds of species. And I've given you some examples here in this list on the left of species that have become extinct due to human activities. Starting way back in 1690, the dodo bird. And what I think is interesting here are some of the reasons that these organisms have become extinct. So the dodo bird were victims of pigs and cats that were introduced into a new environment. The sea cow was hunted to extinction for its fur and its oil. Duck, humans liked its food, shellfish, more than the Labrador duck did. Locust, farmland encroached. Passenger pigeon, there were billions of passenger pigeons in North America prior to humans coming and hunting them, and they hunted them to extinction. The last one died in 1914. Tasmanian wolf had to compete with dogs. Here's a fish. It was the victim of alien fish that were introduced. Here is a species of flower that it went to extinction because of military installations. Golden toad, climate change, and an olive tree just in 2004 was logged into extinction. So how can we try to prevent extinctions? We have to get control of climate change and human encroachments. Humans simply trying to occupy the habitats of species causes them to go extinct. Another thing that people are thinking about is being able to reverse extinctions through genetic engineering using ancient DNA. So for example, you may have heard of Dolly the sheep was cloned from a single cell. So now we know that that's possible. And somebody mentioned earlier that we can get ancient DNA from fossils. So it's theoretically or hypothetically possible to generate a whole new organism from just putting together DNA sequences. And in fact, that has actually been done in the last few years with a bacterium, with using the bacterium Escherichia coli as a host. DNA sequences were put in it and it was made into a new organism. So we know what's technologically possible, but it's going to be far into the future before anything with large species is actually done.
Professor?
Yeah.
Sean Koch here. Is cloning legal? Is there
Cloning?
did they ban cloning after the sheep or?
No, cloning is not illegal. A lot of people do it.
Wait, I think he means by like human cloning.
Human cloning is illegal.
Really? I wonder why.
Because
For ethical reasons.
it would be like the Clone Wars.
Sorry.
Nothing like the Clone Wars.
Yes. Because we must learn from film.
Yeah, well, just to avoid that kind of issue, not approved in any country that I'm aware of, not even in China. Okay, back to the topic. How do we keep track of all these organisms? We classify them, divide them into groups that show relatedness, and then we give each group an unambiguous name that is unique. Now, how is that done? Well, people have been trying to classify living things for a long, long time. And here's an example. Aristotle, who lived in 384 to 322 BC, set up a classification scheme. And I thought you might be interested in how Aristotle saw it. Well, he just looked around and observed characteristics of organisms. And based on that, he divided them into two groups, animals and plants. Well, the plants were easy because he classified them according to size. The big ones he called trees, the little ones he called herbs, and the ones in between he called shrubs. Animals was more complicated. So Aristotle thought that some animals had blood and some didn't. So those that had blood, he subdivided into mammals, birds, non-flying egg-laying organisms, whales, and fish. He distinguished between whales and fish. Those that he said had no blood included things like octopus, crabs, mosquitoes, snails, corals, and anemone, many of the things that you find in the sea. So this and other types of classification schemes existed for a long time until this man, Linnaeus. He was the most influential of all the classical taxonomists. And his criteria for classification are based on the form of an organism. In other words, its morphology and its function. There are a lot of deficiencies in his basis for classification. But the most important thing that Linnaeus did was give us a binomial method of naming things. Binomial means it has two names. Those two names are genus and species. And this is the formal scientific name. So here's an example of the Linnaeus scheme. And the example is humans. So then he proposed this binomial designation for each species. And if you look on the left here, the different designations are kingdom, phylum, class, order, family, genus, and species. So the classification consists of hierarchical groupings called taxa. So each of these is called a taxon. And they range from kingdom to species. Kingdom at the bottom, species at the top. So a kingdom is the largest group, the most inclusive group. And it consists of organisms that share just a few basic similarities, like the plant kingdom or the animal kingdom. Species is the smallest and most exclusive group. You see that one's up here at the top. And it consists of organisms that are similar enough that they can produce fertile offspring. In other words, they're so closely related genetically that they can mate and produce progeny. The genus is a group of closely related species. If we go through this, and for humans, the binomial for humans is Homo sapiens. So they're in the kingdom animals, and these are organisms that are able to move. The phylum is chordates, that is animals with a backbone. The class is mammals, organisms that have fur and milk glands. The order is primates. These are mammals with collarbones and grasping fingers. Family is hominids with flat faces and three-dimensional vision. The genus is Homo, upright posture and large brains, and the species is Sapiens. People with high foreheads and thin skull bones. So this is an illustration of Linnaeus's classification system. So what is the binomial for humans? I've just gone through that. It's Homo sapiens. What's a binomial for cats? Felis catus. Binomial for Shamu, killer whale, is Orcinus orca. Now here's something that is very important and I want you to remember. It's going to be on the quiz on Tuesday. So I have a little note here. Remember this. It's how you designate binomials. When using the name in written form, the genus is capitalized and the species is lowercase. For example, the bacterium Escherichia coli, spelled out like this. The first letter of Escherichia is capitalized. The first letter of coli is not. Both the genus and the species are either italicized, as you see here, or if you can't italicize, you underline as separate words. So you don't have, if you underline, you don't have an underline in the space between the words. So here you see the example, Escherichia coli italicized and not italicized, but underline, no space, no underline between the words. Now, when you're writing, and once used in a paper, the scientific name may be abbreviated. For example, E. coli. So is there any question about this, how you write binomials? Because it's important that you know, and there will be a question about this on the quiz. Okay. Here's an example of writing. Streptococcus pyogenes is a bacterium that's known to cause strep throat. You see the name is spelled out and it is italicized. The antibiotic amoxicillin can be used to kill most strains of S. pyogenes. So instead of writing out subsequent to the first time it's mentioned, instead of writing out the genus name, you just use the first letter and a period, and then the species is written out. So in this case, it's italicized. It could also be underlined.
Can you,
Yeah.
real quick, can you shorten that because you wrote it out first, or can you just have it short without previously writing it?
The latter. The first time it's mentioned, you have to write it out.
Okay, gotcha.
Humans are natural classifiers. So for example, I look at my computer screen, I see all of you, and I just immediately classify you all as students. So think of things that are around you all the time that you classify. You just automatically classify them. You're all sitting, I presume, in a room. You look around the room, what can you classify in your room?
Objects, paintings, books.
Paintings, books.
My
What
bed.
else?
A
Outside
workout
door.
poster.
Furniture,
Yes.
electronics,
Distinguish what room, like the living room or the kitchen or the
Lotion.
bedroom.
right.
Some shoes.
Yeah, footwear,
Games, controllers, this mouse.
exactly.
Piano.
electronics.
Water bottle.
Curtains.
Walls.
Yeah, well,
Ceiling.
you're doing very well, and you got the point, that when we look at things, we immediately, two things are similar, we put them together in the same group. Could somebody turn on the mic? They're getting some background. But since Aristotle, it's been known that many organisms don't fit into simple classification systems, and that's why Linnaeus came along and gave us a more complicated one, but it's much more accurate. And with the onset or the arrival of molecular technology, we now have contemporary taxonomy, which is based almost entirely on the sequences of ribosomal RNA. And I'll tell you about that shortly. Okay, here is a very simplified phylogenetic tree, which points out the 3 domains of living things. So there's a domain bacteria, a domain archaea, and a domain eukarya, which is divided into 4 groups: plants, animals, fungi, and protists. And a protist, we'll talk about a little later, but it's any eukaryote, that is any organism that has a nucleus, that cannot be classified as an animal or a plant or a fungus. And these are usually single-celled. And now, notice we have 2 large domains that are separate from the eukarya. These do not have nuclei. So what's the difference between them? So bacteria versus archaea. Well, first of all, they have a lot of similarities. They're all single-celled. They're all prokaryotes. They all reproduce by binary fission. And we'll talk more about binary fission when we get to cell division. They all have ribosomes and cell walls, and they all have a nucleoid instead of a nucleus. Now, what's the difference? Well, the archaea are what's called extremophiles. Bacteria are temperate. The archaea, they have, the bacteria and archaea differ in the type of cell wall that they have. You don't have to remember that. But do remember that the extremophiles live in extreme environments, like hot springs or salt lakes, whereas bacteria live in temperate zones. None of the archaea causes disease of anything, whereas some bacteria are pathogenic. And finally, archaea do not produce spores, bacteria do. So you might say, what is a spore? Well, it's a single cell. It's environmentally resistant. That is, it will stand extremes of temperature or drought without dying. And they do that because they're dormant. They're not metabolically active. They're always asexual. And they're a reproductive body. That is, under the right conditions, a spore will germinate and reproduce the organism that made the spore. So bacteria have spores, archaea do not have them. So we can sort things out into smaller and smaller boxes. I've already given you 3 big boxes: bacteria, archaea, and eukarya. So if we look at eukarya, it can be divided into kingdoms: protists, plants, animals, and fungi. And each of these boxes can be divided into other boxes. And if we have a complete classification, it's kingdom, first of all, domain, then kingdom, phylum, class, order, family, genus, and species. You need to remember these, and various people have different mnemonics to help them remember, but you at least need to know the sequence in which these classifications occur. Now let's go through the classification of 3 species. Species 1, species 2, and 3. Domain, they're all eukaryotes. That is, they all have nuclei. Kingdom, they're all animals. Phylum, they're all arthropods. Now we get to the level of class, you can see that 1 is a crustacean and 2 are insects. Order, 1 is Decapita, 2 are Diptera. Family, at the level of family, they become distinct. Acaridae, Drosophilidae, and Metacera. The genus and species, now this is the binomial. So the first one, the genus is Homarus, the species is Americanus. The second one is Drosophila melanogaster, and the third is Aedes aegypti. So what are these 3 species actually? Well, Homarus americanus is a crab. Drosophila melanogaster is a fruit fly. And Aedes aegypti is a mosquito. Now, I'm going to give you a sample of my sense of humor. It's based on the fact that the fruit fly, Drosophila melanogaster, is a model organism. It's used by geneticists and developmental biologists all over the world to study the basics of life. So there are many scientists in many labs around the world that study the fruit fly. And for many of those labs, when you walk in, you'll often see a sign like this. And that reflects the sense of humor of Drosophila geneticists. And in one lab that I once visited, there was an additional sign of this type. Time flies like an arrow, fruit flies like a banana, and house flies don't like going flat. So that's my aside for the day. It may make you smile or maybe not. So getting back to business, what criteria are used to classify organisms? Well, classification is based on phylogeny. Well, what is a phylogeny? It's based on evolutionary relationships between organisms. And as I said earlier, the contemporary way to classify things is based on ribosomal RNA sequences. These are highly conserved. They act like a biological clock. So now we're going to talk about wings, phylogeny of wings. Here you see wings on a bird, wings on a bat, and wings on an insect. They all serve the same purpose, flight, but they're different. And here are 2 terms you'll need to remember: analogous and homologous structures. So first, let's talk about homologous. So if you look in the upper left, you compare wings on a bat and a bird. You can see that they both have in their upper arm a humerus, a radius, and an ulna, and they both have fingers, both bats and birds. And if you look at the phylogenetic tree, a very simplified one for the phylogeny of wings, you go back to common ancestor millions of years ago had four limbs. And then the phylogeny splits, and on one side, bats and mice evolved as separate species. The bats evolved their four limbs into wings. And similarly, on the other side of the split, birds and crocodiles evolved. Birds evolved their four limbs into wings, and crocodiles didn't. So what's common here is that the four limbs that started out way back here, where all the animals had four limbs that they walked on, as evolution proceeded, two species, bats and birds, evolved their four limbs into wings, whereas other species didn't. They kept their four legs. And if you look on the right, you can see this again re-emphasized, a bat wing and a bird wing. The bones shown in the same color are homologous. So they're really legs that have evolved into wings. Now, an insect, on the other hand, doesn't have bones. So the supports for the wings are not homologous with the bones in bats and birds. Insect wings have a different evolutionary history. If we then look at definitions, analogous means similar function, but not evolutionarily similar. So if you compare these three, all three wing types have a similar function. They enable flight. But two of them are evolutionarily related and the other isn't. So homologous structures indicate that bats and birds share a common evolutionary past. Analogous structures, for example, a honeybee has a wing similar in shape to a bird wing or a bat wing, and they all serve the same function, but evolutionarily they are derived from different embryonic origins. Any questions about what's analogous and what's homologous?
No, but when you say these animals or beings develop their wings, how did they turn to wings? I mean, what? Because you said they were legs before, and then they evolved to make wings. How did that become wings? Is it some in the air that came together that formed that film around it to make it a wing, or I mean, that the alligator didn't get?
Yeah, function drives evolution. So, and we'll talk about this later in the course, how evolution and natural selection are linked, and they're responsible for all the different life forms that we see. So natural selection is the key driver of life forms. Natural selection favors the form that best fits the environment. So bats and birds are able to fly because being able to fly facilitated their ability to reproduce. So natural selection preserved bats and birds as flying organisms.
Hi, this is Steve Stinson.
Mhm.
So over time, most anything can develop another means for coping with their environment. Pretty much any animal that we know of.
Yeah, and this goes to another point, and that is variation. There are a number of ways that an organism can vary from its relatives. And we'll talk about those. Mutation is one form of variation. So variation is being introduced into populations all the time. And it turns out that some variants are favored by natural selection, and some of them are not. So the ones that are favored are the ones that survive. And the one, when we look around and we see living forms, these are the ones that are able to survive at this slice of time when we live right now. All other forms that can't compete with the ones that natural selection has chosen, they don't make it. They die or never survive. As I said, when we talk about genetics and inheritance, we'll consider a natural selection once again and how natural selection shapes evolution. Okay, the criteria for classification of organisms. Morphology, in other words, the shape of an organism, is the classical Linnaean criterion. More recently, morphology is still considered, but much more exacting is the sequence of ribosomal RNA. So why is rRNA so useful for classification? Well, there's two reasons. One, it's found in all organisms, from the simplest prokaryotes to the most complex eukaryotes. They all have ribosomal RNA. The other point is that some regions of the sequence are highly conserved and some are highly variable. So the highly conserved regions are reference points for molecular analysis. The highly variable regions allow us to discriminate among species. So if you look here at the lower left, this is a molecule of ribosomal RNA. You can see that there's base pairing. We'll talk about this when we talk about molecular biology, but there's base pairing, which gives this molecule a particular shape. And you'll see that in this color scale at the bottom, the red residues are highly conserved, whereas the blue and purple residues are not conserved at all. So here's a region that's not conserved, it's highly variable. Here are some residues that are conserved in every organism. And you can see that in the illustration, the conservation of sequences in our RNA comparing mammals and bacteria. So the top three sequences, you can see the A's and T's and G's and C's. The top three sequences are from mammals, human, mice, and rats. And you can see that all of the yellow residues are similar, and the differences are either green or red. So among mammals, there are sets of green sequences that are common to all mammals and not common to any bacteria. If you look at the yellow sequences here on the right, it looks like they're common to life. All organisms, from bacteria to mammals, all have these highly conserved sequences. Whereas if you look at the actual A's and T's and G's and C's, you can see that there are striking differences between mammals and bacteria at those points in the sequence. So by allowing computers to align one sequence with another, you can look and you can tell immediately whether an organism, whether the DNA sequence came from a mammal or a bacterium. So I hope this addresses the question that you asked earlier about how do you identify organisms, whether the organism is in fact a living thing, or if it's just a sequence that you find in seawater you can still identify what it is from its ribosomal RNA sequences. And based on that, we can come up with what's called a phylogenetic tree, in which you see the three domains, bacteria, archaea, and eukarya. And if you look at how these domains are subdivided, the bacteria have various different categories, and these exist in temperate zones. The archaea exist in harsh environments. Some of them, the thermotypes, exist in hot springs or very hot environments. The methanobacteria produce methane. The halophiles grow in high salt regions. And then look at the eukarya. And I want to point out a feature of all phylogenetic trees. They're all based on lines. And the length of the line indicates relative time, usually in millions of years. So a long distance, like from this common ancestor to this common ancestor, is a long period of time. Whereas these a short line between, let's say, the flagellates and the trichomonads, that's a relatively short amount of time for them to diverge. And what I want to point out here among the eukarya is that the ones that we are most familiar with, the plants, animals, and fungi, are way out here at the tip of this phylogenetic branch, which means that not only are they closely related, but they haven't evolved until just recently. And for most of evolutionary time, we only had these other organisms, which by and large are microscopic, and we don't ordinarily encounter them in our daily lives. What we see are the animals, plants, and fungi, and they are relatively new organisms on this planet. Okay, let's look at bacteria. I think I mentioned before that there are three basic shapes to bacteria: the rods, the spheres, and the spirals. The archaea, they're prokaryotic, single celled, they're extremophiles. They are ubiquitous on Earth, so even though they tend to exist in extreme environments, they actually are everywhere on the planet. Most are not culturable. Their existence is known from metagenomics, which I described earlier. So you can't grow these things in a petri dish or in a culture broth, but we know they exist because they leave their DNA fingerprints in their environment. Archaea are abundant in animals. None is known to be a pathogen, although they are hosts of viruses. And they're intermediate between bacteria and eukaryotes. Some pictures of different shapes of archaeal cells. They're always single-celled. Now, if we look at the domain eukarya, eukarya means what? They have what? The prokaryotes don't have.
Sean here, a nucleus?
Exactly. Eukarya are defined by the fact they have a membrane-bound nucleus. So this includes the animals and plants. The size range of the cells, not the organism, but the cells, are 10 to 100 micrometers and generally divided into 4 kingdoms. The protists I mentioned earlier is sort of a grab bag of anything that doesn't fit in another category is called a protist. And so they're extremely diverse. Here there are amoebae. Here's an organism called Volvox. More examples. Now let's talk about the fungi. Highly diverse. Here's a point I want to emphasize because it works so well for fungi and the habitats that they occupy. Their stomachs exist outside of their bodies. We're familiar with stomachs inside our bodies. Fungi do it the other way. Their stomachs are outside. So how do they do that? They obtain food by secreting enzymes outside of their cells. These enzymes break down any food that is out there in their environment into small molecules. And then the fungal cells take up these small molecules, absorb them as nutrients, and that's how they grow. I want to give you an example of a fungus and how it has impacted human existence. It's the fungus known as Penicillium, and it has impacted humans because it produces the antibiotic penicillin. I want to tell you a little story about how this happened. It's based on a man whose name is Alexander Fleming. He lived in the late 1800s and early 1900s. He discovered penicillin in 1928. He won the Nobel Prize for that in 1945. So here you see him in his lab. This is long after he discovered penicillin. This is a typical microbiology lab of that time with petri dishes, test tubes. And here he's holding a petri dish with a colony of penicillium growing on it. And he's holding in his hand what's called an inoculating needle, which is just a piece of platinum wire attached to a handle. And he uses this to transfer the fungus from one plate to another. Now, up in the upper left here is a picture that Fleming took of the original petri dish in which he discovered penicillin being produced. And I want you to notice I've typed out here what he wrote in longhand. At the top it says the original culture plate on which penicillium was observed. And at the bottom, he wrote large penicillium colony at the top and the Staphylococcus colonies around, showing degeneration. So what had happened is that Fleming had inoculated a large number of plates with this bacterium, Staphylococcus, because he was studying them. And then he was gone for a few days, and when he came back, this is what he saw. He saw that his bacterial colonies were all growing nicely, except he had a contaminant in the plate. The contaminant is obviously a fungus. Now, most microbiologists, in fact, all microbiologists up to this point, when they saw a contaminant in their plate, they said, damn, and they threw it out. But Fleming, on this particular occasion, he had a moment. Not aha, but I wonder why the bacteria aren't growing near the fungus. And he hypothesized, well, maybe the fungus is making something that it secretes out into the culture medium, and it's killing the bacteria. And it turned out that that substance did exist, and it is penicillin. So here is a picture of these are spores. I told you earlier what a spore is. These are spores of penicillium. And here it is growing on a culture medium in a petri dish.
Question. On the
Yeah.
Petri dishes, they use different medium to get stuff to grow, so or they come up with something that works all the time?
There is no medium that works for all organisms, but there are some media that work for a large number. So for example, there are standard culture media for the type of fungus that penicillium is. So you could grow, for example, the genus Aspergillus on the same medium that you can grow penicillium. But usually bacteria don't grow very well on a medium for fungus. In the case of Fleming's original plate here, the fungus, whatever medium he was using, was good for bacteria, but the fungus liked it too. So you know fungi. You've seen them in your refrigerator like this. Some fungi are highly specialized in how they get their food. In this case, I told you that they have their stomachs outside of their bodies. In this case, the fungus is this white thread, and it's made a specialized structure here. It's composed of 3 cells, which is a trap. It's a circle, and it's trapped. This little worm called a nematode has stuck its nose into the trap. The fungus responds to that by clamping down, squeezing the nematode, and then it starts to secrete enzymes which digest the nematode, and the fungus soaks up, absorbs the nutrients, and grows more traps. We benefit from fungi in many ways. Some fungi are pathogenic, cause disease of either plants or animals, but many other fungi are beneficial. So example, several different types of cheese are made with fungus. The yeast, Saccharomyces cerevisiae, is a fungus, and it helps us produce beer, wine, and bread. Now, the largest living organism on Earth, it's not a blue whale, it's not an elephant, it's a fungus. The scientific name is Armillaria gallica. This one was discovered in Michigan's Upper Peninsula. Its body covers 173 acres, which is about the size of the footprint of the Pentagon. Here you see the Pentagon. So that's about the area that this fungus occupies. And the way you see it is through its fruiting bodies. These are mushrooms. You see a large number of them here. Here they're up close. It can grow on dead bark, but it's underground. What you don't see, that there are these little threads growing underground, and that's what you see that underlies an area as large as the Pentagon. So if you, and it's all the same organism, all the same clone of one organism, and if you add up all these little threads, it comes to 440 tons, twice the weight of a blue whale. And the nickname is the humongous fungus. This thing will grow on culture media, and it spreads underground, as you see here, from tree to tree, and makes mushrooms in between. And the mushrooms make spores, and that helps the fungus to spread even further. So this was thought for some time to be the largest organism on Earth. But then we turn to plants. The main distinction of plants is that they're green, most of them. And the fact that they're green, that means that they produce chlorophyll, and chlorophyll is this amazing pigment. We'll talk a lot more about this later in the course, It can absorb light and convert the sun's light into chemical energy. And that is why life on this planet exists, because there are plants. But here we have largest living organism on Earth, 2. What is it? It's a tree called the Pando aspen clone. It's in Utah. It occupies 106 acres. It's about 80,000 years old. And this clone weighs 6,500 tons. So it's even bigger weight-wise than the humongous fungus. So what is the clone? It's 47,000 aspen trees. So they're all individual trees, but they're actually all clones of the same tree. So how can this be? It's because underground, there's a root system that keeps sending up new shoots, and each new shoot is a clone of all the rest of the trees. However, the future of this enormous organism is not good. For these reasons, maintenance of the clone depends on these new shoots growing up from the roots. However, the young shoots are a delicacy for deer, and with humans around, for cattle. So as the new shoots appear, they're eaten. Now in this area, there is no deer hunting allowed because it's a nature preserve. And the predators are all gone. The wolves and the bears have all been eliminated by encroachment of humans. So there's nothing to stop deer and cattle from eating the new shoots. So they simply, the clone is not being replenished. So the lifespan of an aspen tree is from 50 to 150 years, which means that, so that's a fairly short life for a tree, which means that as these trees die, they're not being replaced by new trees. And so eventually this entire clone will disappear. And then just a quick note to be complete about the animal kingdom. There are all kinds of different forms. Here's a little nematode that you saw the fungus eating a few slides back. An octopus, a lot of underwater forms. Here's an insect, and here is a bird. So I think that is the end of this slide set. Yes, it is. And we're just out of time.
I have a question before we leave. This is what's going to be.
Okay.
Yeah, Priscilla. This is what's going to be on the quiz for Wednesday, correct?
This slide set, yes. And I
And
pointed
also
out some
the
of
one that
the,
we saw last Wednesday.
pardon?
Also, so it's this slide set and the one that we saw last Wednesday.
Yeah, and the one before. So there are 3 slide sets, and you'll find them all in your Canvas canister. And you will also find the recording. I've sent you the links for 2 of them, and when this recording is processed, I'll send you its link, along with the pages in the textbook that you should read about classification.
Professor?
Yeah.
Okay, now you said this slide here is going to be on Wednesday's test, and on different one of the slides you said specifically, we're definitely going to be on there. Now, you've given us a lot of different words and spellings. Some of the things I know you can pronounce that I know I wouldn't ever be able to think about pronouncing. Are these going to be where we're going to have to know these actual words and the spellings on the test as well? I ain't gonna remember none of them.
No, you have to make your meaning clear. You don't have to pronounce them on the test. You have to have spelling that's close enough that I recognize what you mean. If I can't recognize it as a word, then I can't give you credit for it.
Oh, shoot. Okay. Can't do like multiplication so we can pick out maybe the word looks like. Okay.
Well, I don't think, at least on this quiz, the issue you're raising is not going to be an issue.
Okay. Yeah, 'cause I'm worried about all the spells with these different words and different spell. It's a lot.
Okay, so just making sure, Professor, it's today's slide and last Wednesdays and Mondays?
Yeah.
Okay.
Professor, I gotta go. Have a nice day.
Okay. All right. I have to go too. So thank you all.
Good one.
And, I'll see you all on Wednesday, and don't forget.
Thank you,
we'll have a
Professor. Have a good
quiz
one.
on Wednesday.
Bye. Have a great day.
Okay.
Bye.
Bye.
Okay.
Bye.
Bye.
Professor. Mike, sir.
Bye, guys.
Okay. Bye.
Bye.